when we started this survey, there was roughly 5,000 of adults that would return to this river every year. You also start to see signs when you gain that much more forage fish in an ecosystem. You'll see things like more ospreys feeding. You see more eagles nesting. You see that, different species like the seals, they'll be up and foraging in these, patches of the estuary that have, this higher number of fish.
Welcome to Shell Phone, the podcast that gives the ocean its very own hotline. Join us as we hear from ocean stewards, discover threats to ocean health, and learn ways we can all answer the ocean's call. This podcast is brought to you in part by the Tampa Bay Estuary Program and Coastal Creative. I'm Harmony Dawson, and joining us this episode is Justin Stevens and Julia Sunnarborg, who will be talking about the restoration and recovery of the Penobscot River estuary. Thank you both for joining me today.
Happy to be here.
Thanks, Harmony.
So first, I would like both of you to kind of introduce yourselves and give us a little background on your involvement in this project or your work in general.
I can go first. So my name is Julia Sunnarborg. I'm a PhD student at the University of Maine, and really broadly, I'm interested in developing non-invasive tools, particularly with environmental DNA, that we can use to study recovering species and recovering ecosystems. So I'm one of the collaborators on this project, along with Justin and with others, and this work supports one of my dissertation chapters.
Yeah, my name's Justin Stevens. So I work for Maine Sea Grant at the University of Maine. And my background in this project goes back a little over 10 years ago when the monitoring in the Penobscot estuary started, continuing through a partnership with NOAA Fisheries. And we're interested in looking at the response of the estuary fish community to upriver dam removals and other conservation measures that are being put in place.
Great. So diving into today's topic, we're going to be talking about the Penobscot River specifically. Could one of you give us some background about the river? That could be where it is, how big this area is, the type of ecosystem that it is, some key species, anything like that that you think is important for listeners to know before we get into the rest.
You want me to take that one, Julia?
You go for it. You got feet on the ground on this river.
All right. Yeah, so the Penobscot River is the largest river that is within the state of Maine. The watershed area covers about a third of the entire state of Maine. River drains into the largest embayment in Maine, which is right in the middle of the Gulf of Maine complex. So that's it's a very productive piece of the northeast ecosystem, part of that northern shelf system offshore. So the history of the river really dates back to indigenous people settling the area and connecting with the river. The tribe that is local to the area is called the Penobscot tribe. They connect with the river in various, physical and spiritual ways. That history is thousands of years old. So the more recent history is Bangor, Maine, which is kind of at the interface of the river and the estuary, became a booming town for the production of lumber and also shipbuilding pretty soon after colonialization. And the river was used to transport logs from large swaths of the state. And then, that lumber got shipped, all over the place. So that was kind of the early history. And part of that, the transport and then the development of, powered mills and things like that, a lot of dams were built across the Penobscot. And going forward, towards more the 20th century, more advanced mills to make paper and things were put along the river, different garment mills. So what ended up happening was, first, the sea run fish that I study were disconnected from the ocean because of these dams. There weren't fishways put in, and very few fish could return from the ocean to spawn up in the headwaters of the Penobscot. And then with the industrialization, the water quality was quite declined and poor. So pollution really was the last piece that nearly extirpated all of the fish in the river. So there were very few areas that could support fish as of, say, the 1960s. So since then, first the implementation of the Clean Water Act and subsequent conservation measures have begun to clean up the river. The water quality in many places is, the highest, the headwaters is some of the highest quality that you can have. So when the fish began to be basically allowed to, survive again, people were focused on the fish passage. So for the past 30 years, fishways have been developed and designed and implemented, but struggles with getting all the different sea run species. Salmon are the iconic one. They're also really good at swimming and jumping, so they can navigate, different types of fishways. But some of the lesser, poorer swimming fish, say like a rainbow smelt, it's really small. They can't ascend, those fishways. Same with like river herring. So back in early 2000s, there was a plan to remove some of the lower main stem dams, and when that was going to occur, it was one of the larger projects in the United States that ever had tried to take out main stem dams. So NOAA Fisheries supported that effort by monitoring a number of aspects of the ecosystem. And one of those was monitoring the estuary, which is this habitat that is interfacing the ocean and the river, to see what kind of response the estuary might have for in response to the dam removal. So all of the sea run fish that are native to the state of Maine were in or around the estuary, Penobscot estuary at the time. So there was hope that by providing better passage that we might see increases in those populations simply by providing more passage. So that was kind of the backdrop to the investigations that we'll talk about more today.
This might be kind of going back over some stuff that you've already said, but in terms of the impact that dams have once they're in place, they impact the movement of the water and the movement of fish. Are there any other major impacts that they have on a river, like sediment, or have we kind of covered all of those already?
Yeah, dams just generally change the flow of water so that they, in the immediate vicinity, all of the habitat that's upstream of a dam is much deeper than it normally would be. So that can change things like the fish community. You have fish that have a tendency to prefer slower water, maybe slightly warmer water temperatures than if it was a run-of-the-mill river where you'd have species that, tend to be like, faster, maybe cooler water. So that's one of the major changes that a dam does besides, restricting the passage of, the organisms.
And then kind of my final background question is you mentioned a couple of the different specific fish species, but what other animal species would you find in this river? Just to kind of give our listeners from different areas of the U.S. or the world a better idea of what you're looking at.
Yeah, so the Penobscot River has the 12 sea run species that I talked about. So those species require both freshwater and ocean connection. But there are dozens of freshwater species that are native to the drainage, small shiner-like species. One of the more popular for fishing is brook trout. They're native to Maine. But then down in the marine environment, we may have fish like the Atlantic herring, which is kind of the primary forage for the Gulf of Maine, a real staple species for, all of the things that would eat them. So larger fish and birds and marine mammals. So in our Penobscot Bay, we have a couple different marine mammals. We have harbor seals, we have gray seals, we have harbor porpoise. And then in the birds, there's a number of, duck species that we have that migrate, through the bay. Our primary bird that preys on fish is the double-crested cormorant, which is they're from Maine all the way down to Florida. So most people are pretty familiar with those birds. But they come up to Maine in the Northeast during the summer because this is where they lay their eggs and they nest, so producing the juveniles that will eventually migrate down south for the winter. We also support birds like bald eagles, ospreys that all live in and near the river and the estuary and rely on the fish in that estuary to survive.
So when we talked previously, you mentioned that your major question in this is to ask how the Penobscot River is responding to the restoration. So before we talk about the findings and what you've seen so far, I'd like to ask about the types of research and data that you are utilizing to answer this question. So you both kind of have your own specialization. Whoever would like to go first, I know we'll be talking about acoustic surveys, eDNA, and maybe some other more like traditional survey methods, but I'd just love to hear more about those.
Yeah, I can kick off to start. When the survey was designed around, trying to observe the changes in the estuary, the team that was involved settled on using hydroacoustics to monitor. And for folks that aren't familiar, hydroacoustics is just generally using, sonar or sound waves that go down into the water and are recorded by a computer back up on the boat to measure the amount of things that are in the water. So there's more technical aspects. We can, measure very, very accurately the signal that comes back, and through that we can infer the types of species that we're seeing when we send the sonar down. More people are familiar with their sonar in their boat that looks at the bottom, and people use them as a fish finder. So they'll see those little marks on the fish finder of their boat, and then they know, that's where they want to fish. So the setup we have is very similar. It's just a scientific brand of it, so that it measures things in a way that's repeatable and much more accurate. So those are the type of data that we'll go and collect from the time that the ice out of the river, which is typically April, and we'll continue the surveys all the way through the summer, fall, until the ice starts to form back on the river in November or December. So, and I guess I'll just add that we, for some amount of the time, we were able to use a really traditional method of catching fish, which is called a trawl. We tow a net behind a boat and we catch fish that we had seen on our fish finder. And then we're able to say, okay, we had this species, they were roughly, this size class. And through time, we were able to develop a relationship between what we saw on our fish finder screen and what we caught on the trawl. So now we just simply use the hydroacoustics data to infer what we're seeing for species on the river. But what got Julie and I together was that the estuary and the bay, it's an ecosystem that's open to the ocean and is constantly changing. So the Gulf of Maine has experienced, the warming event over the last decade, and we've heard reports of changing species complexes. Some are really evident. We have a fish called the menhaden that comes up from the Carolinas during the summer, and summers up here in Maine. And you can see them right on the surface. There's these huge schools of fish. So we knew when we went through with the sonar that we were going through these schools of menhaden. There are other schools that form that we can't see what they are because the water is deep, it's dark, it's very cloudy. So the sonar sees the fish, but we can't. So, and we weren't able to do that trawling anymore. So we had this question of exactly what were these, like single menhaden that were around in the schools, or were they some other type of fish? So that's when we partnered with Julia's lab to try to implement a new technology or a novel technology that might help us to answer that.
Great. Before we move on to Julia's piece of this, I'm curious what the reasons for moving away from the trawling were. I mean, I can think of a couple off the top of my head, but I don't know like your specific case, and maybe it would be helpful to just share a little bit of like why, and then like what impact like that trawling has on like an ecosystem, and like whether the risk outweighs the benefit or not when using it in science. if that makes sense.
Yeah, it's a great question, Harmony. And yeah, to be blunt, so trawls are a fairly invasive type of sampling. You have to capture the fish in a net, and they all go back to the end of the net. And when you pull that net up into the boat, they're restricted, they're crushed, and they don't have any more oxygen. Right, they're not in the water. So typically, when trawls are used, it's a process where all of the fish are killed. So it's a designed net for, say, commercial fishing, where you're not just going out there to catch fish for fun. You're catching them to bring back to shore and sell them at fish markets. So that's the same device that we had used, because traditionally in fishery sampling, that's that's what's used. It's been around for the longest amount of time. In our estuary, we had to deal with a number of factors that made it less effective to trawl. One, we knew that we were interacting with endangered species. So the Atlantic salmon, we could catch every so often in the net. We had to ensure that we weren't damaging or killing those, because we're working to protect those. We don't want to do more harm. We also have 2 species of sturgeon in the river, in estuary, the short-nosed sturgeon and the Atlantic sturgeon. We didn't want to capture those to, again, as researchers, our motto was basically do no harm. We don't want to do worse for the fish in order to collect information. The other thing is that trawling is really expensive. So you have to pay for the boat, pay for large crews to collect that data. Whereas, with the hydroacoustics, we were able to still accumulate the same amount of information, just not at the scale. So we didn't have that species-specific detail that we had when we trawled, but we had enough of that trawling information to go forward with, our findings and to evaluate, changes.
Great. So that leads us into Julia's role with eDNA. We've heard a little bit on this podcast of what eDNA is, but maybe if you could just re-explain that a little bit, and then tell us a little bit about how you collect it, how you analyze it, what it tells you in this case.
Absolutely. So eDNA, or environmental DNA, is just this idea that animals and organisms that are living in an environment are leaving behind traces of themselves. So DNA that comes in cells from feces or skin cells, just kind of floating around in the environment. And we can capture this environmental DNA and learn something about who is living there and what they are potentially doing, without ever having to catch a fish. As Justin said, it offers a way for us to learn about an ecosystem without directly taking from the ecosystem or harming it in some other way. And so this is a fairly new tool that's been developed over the last decade or so. And so there's still a lot of things that we are learning about how the tool works, what information it can provide, and the role that it can play in ecosystem monitoring and management. And so the goal of this project was to kind of investigate how this could help us understand this system. And the system is a really great one to learn about this tool in, given that Justin's been working on the Penobscot for the last decade plus, and we have a lot of information about the fish that live there and the tools that we're using. And so it's a really great place for us to add in this new technique and learn about it and use it to gain some information. As Justin mentioned, not having the trawl anymore, there are instances where it's hard to understand the exact species. And that's something that eDNA can help us with. So we started incorporating eDNA sampling into Justin's hydroacoustic survey. And so when he goes out and runs his survey along the estuary, he collects a couple bottles of water for us at some predetermined points along the estuary. So it's very simple from its collection method. Justin can I gave him a cooler full of bottles that are empty, and he can just dunk them in the water as he goes along. So it's very simple from a collection standpoint. There are more complicated ways of doing it, but at its core, it can be really accessible. And then that water just comes back to the lab, and I filter the water, which essentially just captures all the floating particles on a filter that can then be extracted and stored at the University of Maine. So once we've kind of captured that genetic material, we can ask a couple different types of questions about it. We can ask how much DNA there is for a particular species, which is helpful for thinking about, how many fish are there and when are they most common. We can also do DNA sequencing, and we can actually determine the actual sequence of letters that DNA is made up of. And that allows us to ask questions about which fish are there, which animals are using this environment, and when are they around. So we use both of those techniques in this project to ask questions about if eDNA can do things like track when a species is migrating, or what kind of questions we can ask about the community as a whole. So can we get that taxonomic information that we might not always be able to get from just the acoustics alone, especially when you have fish that are really similar to each other? How can we kind of like expand our toolbox with this new method? Which has been a really exciting place to experiment and a really fun way to interact with this ecosystem.
Yeah. I have one like smaller kind of random question, and then I have a more like broad question about that. So the first one is, when you collect the water, is there like a certain depth that you get the water from, or does it matter where you collect the water from in the water column?
Yeah, so for this project, we collect, and by we I mean Justin collects water from pretty much the surface, so just under the surface of the water, the way you could reach in and grab it from the side of a boat. There are ways to collect water from deeper in the water column, and some projects do want samples from certain depths. It depends a little bit on your environment. If you have water that's mixing around a lot, then it doesn't matter quite as much where in the water column you sample from. But if you're in something like a stratified lake, maybe you do care if the fish are at the bottom, maybe you want to sample at the bottom. For in this project, we are assuming some level of mixing, and we get a lot of DNA from the surface. Does a pretty good job of representing the system. So ours are collected just under the surface of the water.
Going back to about how part of the purpose of using eDNA in this case was to kind of see if it could be an effective method in studies like this, and like what kind of information you could get from it. You might not have a simple answer to this question, but do you feel like that has been successful so far? Do you see eDNA having a major role in these types of studies in the future? What's your general feel about that?
Yeah, so we went into this, at least I went into this, as pretty an open mind about what we would get back in terms of information. And I've been really excited and really impressed by what we can get back. So we've seen that the amount of DNA in the water is reflecting, let's say one of our fish species is migrating, the amount of DNA in the water is peaking when the fish are migrating. So it is capturing these aspects of life history that we know exists in the water, which is really exciting to see that line up with what we would hope and expect to see. We've also seen that by doing our DNA sequencing approaches, we can detect the presence of a really wide variety of species. So Justin mentions that we have like 12 sea-run, like migratory species that are using the estuary on their way to and from freshwater in the ocean. And we can detect all of those species in our water samples, which is exciting. We also detect a variety of freshwater species. We detect marine species. We detect ducks. We can detect seals that are using the estuary. So the breadth of information that we can get is very exciting in terms of effort that we have to put in to get that information. It's cool to have another layer that can provide some of this information in a system like this. So I have come to really appreciate the role that it can play, especially when it's aligned in a system that has other tools being used. Like Justin's information that his survey provides about fish biomass is absolutely essential. We can't get that from eDNA alone, at least right now. And so having two methods that work really well together has been a really cool way to get like a more holistic picture of what's happening in the estuary. So it's cool to see the ways that it can support like these ongoing efforts. And it's cool to see the ways that ongoing efforts can support how we understand this tool.
Yeah, great. This is a very broad question, but as I'm in a research methods class right now for environmental studies and was reading through the resources that you guys sent over to me about this project, I wanted to ask what some considerations are that go into designing a scientific study like this, or just thinking about it in general.
Yeah, I can speak to a little bit from the perspective of someone who is in graduate school right now and is kind of learning how to come up with and design studies. So for me, this project was originally not part of my dissertation, but as I was working on other projects, I had questions that kept coming up about this tool, and I had a lot of things that I wanted to test. And we've been really lucky in that we've been working with Justin for years, and there's a really beautiful infrastructure of efforts that are being done on the Penobscot that provided for me a really cool place to test some of my questions, and kind of refine this tool for, many broad purposes. And so for me, it was kind of born out of a need and out of a lot of like blossoming questions that I had. And I got very lucky to have an infrastructure to do that in. And so then for me, it became a question of how can we best target our efforts to answer these really specific questions. And there's always limitations when it comes to things like resources and funding and time. And so we, collect as many samples as we reasonably can. And we are lucky to have a lot of information about the system that helps our study quite a bit. And so we, the system is set up in a really nice way, and that Justin, has a seasonal window that he covers with the survey that works really well with the questions that I wanted to ask. We have resources at the University of Maine to do a lot of our eDNA methods on campus. And I have funding to ask these questions. And so those components kind of come together to set up kind of what you can achieve. And we've gotten really lucky here to have a lot of awesome people involved in this project.
Yeah, great. Was there anything that you wanted to add to that, Justin, before we moved on?
Yeah, I'll just echo that it's been a really exciting project. From my perspective, you study fisheries and you learn all these kind of classical techniques to sample, and you don't necessarily learn that they're all trying to do the best at capturing what's actually happening in the environment. But fish and wildlife are inherently unpredictable. So their behavior, the different environment response to environmental parameters that they may exhibit all impact your ability to sample and the interpretations of your samples. So fish are a great example. Some of them really like to form schools. So if you're sampling and you catch a school, great. It looks like there's a lot of fish. There might not be a school right next to that school. So then you sample and you don't catch any fish. So we talk a lot about in the fisheries world about the distribution. So like where the fish are. And that inherently sends you down this path of thinking where they are at what time of day or what time of year. And I have always been fond of the hydroacoustics technique because you can look at the water column beyond where the nets can reach, and you can do it continuously because you never have to bring the net back on the boat. So any of that time you're cruising along, you're able to kind of see what's in the water. The downfall, there's downfalls with it though. So there's been, sometimes the schools of fish kind of know a boat's coming, so they get out of the way. So then you're not able to see them on the acoustics. We mentioned a lot about kind of identifying the species. So this technique is really promising in that the fingerprints of those fish, right, that the bits that are coming off in the water aren't going to be susceptible to those same things. They shouldn't. The particles of DNA aren't going to avoid a vessel per se, right? And that patchiness should be accounted for a little bit. It's also exciting because Julie and I have talked a lot about how there's a lot of unknowns. So I mentioned, that we're in an estuary, but just for folks, if they're listening more from Florida, so our tides range around 14 feet every tide, up and down. The river, fresh water that's coming out of the Penobscot is, on average about 15,000 cubic feet per second. So that's, I can't remember the conversion to gallons, but there's a lot of fresh water coming down into that tidal area. The mixing that happens is tremendous, but it also varies. So, that's why when we designed this, we said we better take more samples than we think, cover some of these differences in, fresh water and brackish water and marine, and do it for the season, because we're not sure how those things may, how the DNA might distribute differently depending on what time of year it is. So to be, kind of involved with a study that's helping, and will help others when they're designing their studies, I think that's really exciting.
So going back to that big question of how the river is responding to restoration projects, what changes have you guys seen so far in the ecosystem, and any impacts as well on like economic benefits, maybe related to fisheries, but really focusing on changes in the species and the ecosystem as a whole?
Justin, I'll let you take this one since I think you've seen this river change
Yeah,
for longer than I've
it
been
was
here.
amazing to see the start through up until this past year even. Um, the response of some species has been very visible. So river herring are typically a fish that they lay a lot of eggs, they make a lot of juveniles, and a lot of juveniles go out to the ocean. So when we started this survey, there was roughly 5,000 of adults that would return to this river every year of the river herring species. Last year, 5 million adults came back to the river. So those multiple orders of magnitude in that one species is visible in the estuary. So what I see on my acoustics is that there are more of these fish in more of the areas of the estuary last year than there were than in the first couple of years that we started. You also start to see signs when you gain that much more forage fish in an ecosystem. You'll see things like more ospreys feeding. You see more eagles nesting. You see that, different species like the seals, they'll be up and foraging in these, patches of the estuary that have, this higher number of fish. So I can just observe that we've seen these changes. Of course, we're scientists, so we want to be thorough in our investigation. So we've actually tested some of these, numbers. Like, is there more fish biomass? And the result is that there is. Is there more birds that feed on fish? Yes, there is. So, it isn't just the observations, it's also the scientific investigations that we've been testing that show that we've seen, a response. So generally, it's characterized as we've got more fish that are in this habitat, and they're the type of fish that support things that are higher in the food chain than themselves, like the birds and the seals. So
Cool to hear about the connection between the fish and all the other levels of different types of animals that are kind of impacting each other. And that kind of leads me into one of the next questions that I wanted to ask, which I got from looking at one of the resources that you sent over, and it just kind of stuck out to me as something that was really interesting. It was talking about how the restoration was impacting the issue of seals preying on endangered Atlantic salmon. And I'm not sure if I'm really talking about that correctly, but I thought that was interesting. And if you guys could talk a little bit about that, that would be great.
yeah, that's so it's been kind of easier to characterize some of these numbers, right? Like how the fish populations have changed because we can count them. What you're mentioning is kind of generally this theme of like an ecosystem process change. So like how are, different parts of the ecosystem interacting as there's changes in their abundance. We're really just starting those investigations. That was the first, the paper I sent was the student that was just before Julia in the same lab at UMaine. She was interested in the counts of seals that we had. And something that has been tracked for some time are the amount of salmon that come back to our rivers that have bites on them from seals. Really any injuries, but seal bites are a particular injury because seals have canine teeth, and those canine teeth leave a particular mark on a fish when it's unsuccessfully predated. So the fish that survive that bite, we can observe this kind of mark on the belly of the salmon. And it's always been a topic in the Penobscot River because people would see seals and they would see salmon that have been bitten by seals, and they would equate the two as being, something that was negative, a negative impact. The seals are negatively impacting the salmon. anglers back in the day would fish for salmon down near the head of tide and would interact with, seals in that way. So this happens a lot in the world, is that, when there's a human interaction with a resource, they can interpret whether there's another kind of interaction. So depredation is a common theme across all the coasts. An angler has a fish on a line, and then some larger animal comes and takes the fish, a shark or a seal. So that kind of spurred an investigation more than 10 years ago at the trapping facility where the salmon were being caught and measured and counted. And at the time, it looked like there were more seal bites than when the time series started. So what Julia's predecessor did was evaluate like the last 10 years, as we've seen all these changes in populations of fish. Has there been a change in the number of salmon that appear to have seal bites on them? So the result of that was that there are less salmon that have seal bites on them now, which aligns with the hypothesis that if there are more things to eat in the estuary, salmon would never be that abundant just because of the type of fish that they are, as compared to a herring, which is traditionally called a forage fish. There's just always a lot of them in big schools. So if you're a seal, you're more likely to run into a river herring than you are a salmon, especially if there's a lot of river herring. So that aligned well with that hypothesis, and we'll be investigating a few more of those and trying to crunch the numbers to understand better what these kind of long-term changes might mean for the ecosystem and these relationships, the predators and the prey, because of course seals populations can go up and down for a number of reasons. So in the northeast, we've seen seals that are susceptible to avian flu. So, we've seen some unusual mortality events related to that. So their populations have been going up generally over the last few decades, thanks to conservation measures like, folks aren't out, removing seals from the ecosystem anymore, which was a common practice up into the 1970s in Maine. You could take seals out of the ecosystem. So yeah, it's a really interesting field. It's also a very difficult one because you're working with large ecosystems. You're not in a lab. There's a lot of variability that gets introduced. So it's something you have to do over a long time. So that's why, we're almost, 12 years, into this investigation, and we're hope to continue for a number more.
I'll just add one thing, Justin, what you mentioned is this idea that if we restore, so river herring have been, like many others, like depleted, and this idea that they might have lost some of their like services that they're providing to the ecosystem. And one of those like theorized services is that if you have a lot of fish at the same kind of trophic level and size, and they're all around, then you can help reduce the predation pressure on things like endangered Atlantic salmon. If there are river herring that we have really abundant, and a seal can go for that instead, that we can also have added benefits for things that really need it, like our salmon. Which is another cool thing to see and to look for as we start to see these like populations recovering.
Great. Yeah. I would also like to ask you guys a little bit about fishing and fisheries in Maine, specifically in this area. I know fishing is very important economically and culturally throughout Maine as a whole. But in what ways has that come into your study, either in executing your study or just thinking about the impacts of it, or its impacts on you?
Yeah, I guess I could take a stab at that question. So there are a number of really important commercial fisheries, as well as recreational fisheries, and I mentioned in the beginning with the intro is the cultural traditional uses of fish in the for the Penobscot people. So the project that we're working on really seeks to benefit the entire ecosystem, which is all of the people that are involved with it as well, both whether you're a commercial fisher. Yeah, I'd say the biggest thing that I notice from folks is that when I talk to them during the surveys, I might stop at a dock that has, recreational fishermen on it, and they'll mention that there's more forage. They're like, there's more bait, in the water. So I think people are gaining, kind of a sense of what the system, used to provide, and what it can provide. But I mentioned that long history of pollution and degradation. There's also folks that still say, like, I can't believe that you're hanging out in the river, like it's so polluted. So there's a challenge to, the conservation side and as scientists to try to get these messages across to people that, one, there's been great improvements, to the health of the ecosystem, and, two, that's going to provide, various opportunities for folks. So the state of Maine and its fisheries management, it is complicated because it's really species specific on, how, say, management limits or, catch limits are set, how many of what species, what size and things, that you can fish for. So it'd be pretty complicated to go down through all of the specific examples, that impact it. But all of the sea run migratory species, unless they're endangered, are at least recreationally fished. So there's that strong connection.
I was only going to say that as someone who doesn't spend a lot of time out on the water, I have had the privilege a couple times of going and watching the runs. So when the fish are migrating, you can see them as they make their way upstream. And there's some cool places along the Penobscot where you can go and you can observe a stream that's just chock full of fish in a way that, as someone who's not from Maine, I had never seen anything like that before. When you go and you can observe those runs and you can see people interacting with these runs or fishing, and like you could almost stick your hand in the water and grab a fish. And it's just like a pretty amazing way that people can interact with these species. And it's been really cool to see that as they become more and more abundant, there's more ways for like the communities to kind of rally around these runs, like culturally, as well as like the economic benefits of having fish available for catching, which is a pretty amazing thing to watch.
We're starting to see public festivals that pop up to see the fish runs, where these streams, where it's, viewable, we can see literally the river, full of fish. And that's really helped us, be that entry point. So us being, folks that are, out there doing the science, like collecting this information and trying to get it back out to people, those kind of social events are a really nice, nexus and a place that we can reach a lot of people and talk about it, because it's kind of this constant struggle that folks are like, they perceive, we all do this, we perceive the world around us, and we think that like the condition that we're seeing right now is something that has always been. So if you hop along a river that is full of river herring right now, you may say, wow, like I'm glad I stopped, but it's been like this forever, right? And the reality is that you're seeing something, like Julie and I are really close to this one stream that was blocked for 250 years. So the last 5, it has been full of fish. But the previous 245, there wasn't a single fish. So, to try to talk about the magnitude of change for folks and give them a perspective, that there has been major change in a very short amount of time. And in the messaging, like I said, there's things that are occurring, that aren't great. In Maine, we're dealing with things like, sea level rise and increased flooding from storms and things like that. Like across the country, across the globe, folks are, concerned about changes. And this is one of those changes that's a positive. And it's hard to describe in words, but when you have a group of, children and parents and families and things, and they're observing a mass migration of, nature, like these fish can provide, it's very transformative for them. And you can read about it in a book, you can see the graphs on a paper, but there's something by immersing yourself in all of the senses, when you can see, smell, hear, and touch them, that really is effective in at changing a person's perspective. So I just say to folks, try to find your local fish run where this can, where you can see this type of thing, because it's a really positive experience.
Yeah, thank you for bringing that up. That is something that we've touched on in a couple of past episodes of how impactful it is to have like a hands-on, real experience that really sticks with you and kind of gives you a point that you can build knowledge from, but also helps you to care about different issues. That is all I have for you. Thank you so much to both of you for joining me today. It's really
Thanks so much, Harmony, for having us.
great to learn about.
Yeah, thank you so much. This has been a real pleasure.
Thanks for tuning into Shellphone. To learn more about our projects, suggest a guest, or donate to the show, visit our website at breachthesurface.org. You can find us on social media at Breach the Surface or at Shellphone Podcast. To learn more about Julia and Justin's work, check out the links in the episode description.